Okay, so good afternoon, everyone. My name is Vanessa Duran, and I attend to Mather High School. I will be a senior this fall, and this is my second year working in the Plant Science Center lab in the soil lab. And my project is soil aggregates. So some background research. I worked with two different types of soils. I worked with soil from prairie redmonds, and prairie redmonds are just pre-settlement native plant communities that have survived on a site to the present day. I also worked with soil from prairie restorations, and prairie restorations are just to restore some of the prairie land that was lost to farming, industry, and cormorants. Soil aggregates are just soil particles that bind together, and water-stable aggregates are just resistant to raindrop impact and water erosions. Macro aggregates are particles that vary from 8 micrometers to 2 micrometers in diameter, while micro aggregates are particles under 2 millimeters. And also a side note, the greater portion of macro aggregates in soil indicates better soil quantity and hence resistant to erosion and enhanced carbon holding capacity. So my question is, does aggregate structure vary within prairie sites, and is there a difference between prairie restorations and prairie redmonds? My hypothesis is that aggregate structure does vary within prairie sites due to soil heterogeneity, and there will be a difference between prairie restorations and prairie redmonds because prairie restorations have fewer macrogates than prairie redmonds due to their history of agriculture. And this is my experimental design materials. So for my experiment, what I used was sieves with different sized screen pores to sort soil from redmond and restored prairies into the various different soil aggregate size categories. And just a couple lists of my materials. So for my method was I weighed 100 grams of soil, and then I poured it into a tin labeled whole, which was used for the soil dry weight. And then I weighed another 100 grams of soil that was used for sieving. So then I grabbed my 4 millimeter sieve and I put it in the tub. I filled the tub with water and I dropped my soil and I just basically sieved vertically, horizontally, up and down, carefully. And then whatever the remaining material that was left, I just poured it into the 4 millimeter tin. I put that in the oven so any moisture and everything could just leave. And then from the tub, I poured my slurry into beakers, and then I placed my new sieve in there, which was the 2 millimeter sieve, and then I poured the beaker and the slurry that was in the beakers back in the tub, and I just repeated the process for the 250 and the 53 micrometer sieves. And it's just a picture of me pouring the slurry into the 2 millimeter sieve, as you can see, and these are my 4 sieves. Again, I used 4 micrometers, 2 micrometers, 250, it was macro, okay, wait, I used 4, the 2 were 4 millimeters, 2 millimeters, 250 micrometers, and 53 micrometers, sorry. And that's just another picture of me, and there's, with me. Okay, so then as you can see, the slurry's in the tub, and then that's my 4 millimeter sieve, and that's just the sample that I grabbed from the sieving. And those are just some samples in the oven. So my data and results. This is the graphs that show the variation within prairie redmonds. And then that's the prairie redmond A and then the prairie redmond B, which are 2 different sites, but within those sites there's 5 different samples. And then the ones on the right are the variation within prairie restoration A and restoration B. So as you can see, there appears to be no significant variation in aggregate structure within prairie sites, within the the 1, 2, 3, 4, 5, but between the redmond and the restoration, obviously there's a difference, as you can see in the next graph. These are the redmonds from all the A's and all the B's and all the 1, 2, 3, 4, 5s that I did. So there's definitely a difference between the renmin and the restoration, and it appears to have a greater portion, the restorations, of macro aggregates than prairie renmins. My conclusions: my first hypothesis was not supported. Aggregate structure does not vary within prairie sites because prairie soil within each site is relatively the same. My second hypothesis was supported. Prairie restorations appear to have a greater buildup of soil macrogates. And then prairie restorations, restored prairies may be more resistant to erosions. They may also hold more carbon than renmin prairies. This is surprising and may be due to differences in management history between renmin and restorations. Some further research should investigate the effect of different management histories on aggregate structure and soil properties of prairie renmins and restorations. This is my works cited and acknowledgements. Just a great thanks to all my supervisors, coordinators, and mentors. Corey, thanks so much. And Lauren, I don't know if she's here, but oh, there she is. Thanks Ben, Shelly, and of course all the donors. Thank you. And Louise, thank you for letting us use your lab. I hope it wasn't such a mess with all the soil and everything. Thanks everyone, and okay, my peers, thank you guys. I'm sorry. I hope you don't. No? Yes.
Well, so you're saying that things that have been restored, that people have gone in and fixed things up, had better soil.
Yeah, I mean, what I was like just investigating was just like, is there like within the site and then within like the within like restoration and renmins, but I'm not really working on like how they could. I just worked if there was a difference, but obviously if like there would be like, I don't know, ways.
Do you think that the remnants were affected by their surroundings, or?
Probably. I mean, obviously it would indicate to be like there should be more research done and more, because I just did 5 samples and then there was the A and the B, and there was actually more samples to be done, but I couldn't get to that because of the short amount of time that I had. My mentor Corey is actually doing that, so she will like take it beyond to what I just did. I just did like a little portion of it, but of course, if we would do more research, we could get better results and like a better idea of what we're dealing with. Any more questions? No?